Thank you so much for that introduction. So I'm really, as Aubrey said, my name's Kennedy. I was mentored by Michael Rose at UC Irvine. A lot of people here probably know of Michael Rose. He's famous for coining the term antagonistic pleiotropy. And I'm really excited about this presentation because, as many of you know, he selected for the long-lived Methuselah fruit flies, and this is one of the first times that some detailed genomic information about them has actually been presented. So for those of you who don't know, the Methuselah flies are the result of a selective breeding experiment that started back in 1980, just before I was born. And so Michael started those at UC Irvine. It's been going on for over 400 generations now, like 44 years. It's just the result of continued selection pressure, taking the longest lived flies, taking only the eggs from those longest lived flies, using those for the next generation, and so on, and so on, and so on, and so on. The original Methuselah flies, which we call the O flies, have an average lifespan of about 70 days. I took over care of the flies in about 2007 and have continued to apply selective pressure to that population. And we now have an average lifespan of about 135 days in a population that we refer to as the super Methuselah flies, or the SO flies. So we have these two populations that live 2x and 4x respectively in comparison to our control flies, which we refer to as the B flies. This is a survivorship curve of the female populations. Just a little disclaimer, this data is from 2020. We are due for another longitudinal survivorship study. It's getting a little bit more difficult to carve out time to do these because they live so long. But you can see between the Bs and the Os, and then the Os and the SOs, between the Os and the SOs, there's an additional 13 years of selective pressure that was applied here. And so this is the result from 2020's longitudinal study. And you can see that the survivorship curve of the super Os, the super Methuselahs, is starting to kind of flatten out. We do have a survivorship curve for males as well. It looks very similar. Their average lifespan's a couple days less, but to conserve slide space, I just put the females up here. These are the daily death counts. Again, for females, the daily death rates for the super Methuselah flies are much lower than the other two populations, as one would expect. So in looking at the variation count of these mutations of the genomes, the Methuselah flies on average have about 20% more mutations than our bee flies. Interestingly enough, the original O flies have like 22% more, whereas the super O flies have 20% more. So so that suggests that there's like more ways to be a moderately long-lived fly than there is to be an extremely long-lived fly, which is interesting. Or perhaps maybe the super O flies have honed in on like the ideal mutations or ideal efficiencies for extreme longevity. There's more. Obviously more studies planned to see why this is exactly. But we have our mean S&V counts compared to our reference genome that we use there in the corner. Also, what's interesting about this, about the 20% factor, is that when you look at supercentenarian humans, it's very similar. So this is from a data set of 30 supercentenarian females compared to normal civilians. So you can see those compared to a Drosophila genome, the mutation burden is roughly the same. In a PCA analysis of the transcriptome levels shows really nice population differences. We have highly differentiated populations. You can see there's couple of super O populations that are very super O, but overall we have 3 very nicely differentiated populations with different lifespans. In a functional analysis of differential expression in gene ontology categories, we found that O's tend to have greater representation than B's in carbohydrate and amino acid and nucleotide metabolic processes. SOs have greater representation than O's in respiration, ATP metabolism, and ion transport processes. SOs have greater representation than B's in aerobic, respiration, and mitochondrial processes, but less in processes related to DNA replication and cell division. Now, this last point, we're kind of unsure because the data that was collected for this last analysis was collected 1 week post-eclosion, so coming out of their pupae for all of these populations, which for our baseline flies, for the control flies, equates to like roughly like, a quarter of the way through their lifespan, like mid-ish life for them. Like it's later in their lifespan, whereas like the longer-lived populations, that's very early in their lifespan. So that might affect what we're seeing in terms of like the DNA replication and cell division expression levels. Interestingly, or maybe not interestingly enough, as we've evolved the Super O flies, what we've also seen in their development, so going from egg to pupae to eclosion, they actually take an entire week longer to develop than the other fly populations. That's something that has developed over the last 17 years that I've been taking care of these flies and selecting for them. We did an analysis of the transcriptome factor activity and expression differences using NetAct, accounting for TF activity of individual samples and inferring gene expression and target genes. This is what that heat map looks like. I know Ben showed this in his talk Saturday morning, but what we saw is a really nice demarcation between the different populations, zeroing in on a few very specific transcription factors that differentiate our super long-lived population from our baseline population, and then the inverse of those expression and activation levels indicate our control populations. There's some familiar transcription factors up there, and we are really excited to look into these further. Some studies that we have planned for the future include specific time point analyses, like time point analyses throughout the lifespan of these super Methuselah flies during our next longitudinal study, pre-reproduction, post-reproduction, early life, extreme late life, seeing how their gene expression changes throughout their life, seeing if there's any changes in this heat map as they live. We also made this gene regulatory network simulation based on the activation pattern. This is just like the picture of this little rubber band ball, but we do have a interactive version of this where you can actually pull on the nodes and see how the activation patterns affect the different transcription factors, and so that they are all connected. So our next steps and further research that we want to do some mitochondrial analysis and expression of phenotypes, some advanced machine learning and AI analyses like supervised mixed omics methods, and using our bioatom space knowledge metagraph that Ben was talking about earlier on Saturday for advanced pattern matching. Longitudinal studies, like I said, to further elucidate this advanced long-lived phenotype. We also want to do some integrated multi-omic modeling using OpenCOD Hyperon, which is the neurosymbolic AI framework that's being developed by our partners at SingularityNet, allowing us to integrate findings from genetic and transcriptomic data and cross-organism data sets as well to see if we can identify more crucial longevity pathways that we can port over to humans and find actionable targets. So with that, I just wanted to say thank you for listening. I have an amazing team. I just want to highlight my Chief Science Officer, Mike Duncan, is actually here. He did most of the analyses in here. I basically just made sure the flies were fed and sucked out their DNA and RNA, and now I'm communicating the results. If anybody wants to talk more, I'm around, and we are also in the middle of a seed fundraising phase for our AI platform that we're also building, and then our fly work as well. So if anybody's interested in that, I'm free to talk about that as well. So
thanks very much, Kennedy.
Thank you.
And we've got time for a question or two, if there are any. It seems like, oh no, there's one at the back.
There we go. Oh, it's Jose.
Fantastic presentation, Kennedy. And what do you see coming in terms of extending even longer the life expectancy of the flies? Is there a limit you see coming up or not?
So far, I haven't found one. Like I mean, now it's the generational extension because the generations are getting so long. I'm only able to extend it by like a few days each generation because now there's 1.5 maybe generations per year in these extremely long-lived flies, whereas I used to be able to get like a couple of weeks gain with each generation. But so far, as long as I keep their cages clean, I haven't really hit a limit. So we'll see.